just remind you a little bit about amino acid. So this is from your biochemistry and molecular biology. So you know we have 20 amino acids. Most of us use the 20 different amino acids to synthesize protein, although there are some exceptions. Then in each amino acid, we have amide group, carboxyl group, and the side chain. So use that can classify the amino acid into different subgroups, and that also affect how likely the change will exist or will remain in the population. And we'll talk about that later when we talk about the sequence differences. We also use certain terminologies to describe the protein sequences. So when we say protein, in your mind, there is a stretch of amino acids they connect together. After the translation, then that's we call protein. But people also use peptide, or we say polypeptides. So polypeptide is very similar idea, but it's usually more than two amino acids connect together. We can call it polypeptide. And in your textbook, there's also definition for polypeptide, and sometimes we use polypeptide and proteins interchangeably. But just make sure exactly what the author means. But residue is something we use more or less the same definition. So in a protein sequence or in a polypeptide, we have each individual amino acid. Then for one particular amino acid, we can say it's a residue. So for DNA sequence, we say it's a site. So when I say there are 20 sites of the sequences, then we'll say in protein, we'll say there are 20 residues. So that's, and we're concerning about like a particular residue, whether that one will affect the protein structure or the protein function. So we don't use residue for DNA, but occasionally you see people do that, but I don't think that's a common use. So we only use residue to describe the amino acid site of a polypeptide or of a protein. Then the other two is relatively easy for you. You just describe it's an N terminal of the polypeptide or the C terminus of the polypeptide. And this is just a cartoon to show you, and this is probably very easy for you to recognize and remind you about a polypeptide. And this something just like a textbook version tell you a polypeptide doesn't really function as a long polypeptide chain. The protein has to be properly folded. So it's not like a long chain of like an amino acid sequence as the functional unit. So the polypeptide has to be folded properly, and you form a sort of like a three-dimensional structure. Then, or some polypeptide or some proteins will have different subunits to combine together then as a final functional unit. So this is sort of like it reminds you about the structure of the protein. And I think the important part for the protein structure nowadays is when we talk about synonymous and non-synonymous side. Actually, we're going to talk a little bit later in this lecture. But the important part is when we talk about non-synonymous substitution, and that is to say the changes at the DNA sequence doesn't affect the protein. And classically, we think this type of site is like a neutral site because the change won't affect the protein sequence. But just very recently, we found this type of change occasionally will have a phenotype. So that is something now we discuss how likely the synonymous substitution is really a neutral substitution. So we just talk about the pseudogene, the same idea, because the pseudogene doesn't affect at the phenotypic level. So we think the site of the pseudogene is under neutral evolution. And the site at synonymous sequence, we also assume, I use assume, there are silent mutations. Those words will come up in the later part of this lecture. But occasionally, we do have evidence to show the silent site is not really silent. And one of the examples is the change affect protein folding and have a mutant phenotype. So this is something concerned as a silent site effect at a phenotypic level. But in general, most of the silent site doesn't really change the protein's sequence or even its function.